Hello, Kasia. Hello, everybody.
Hello.
Can you hear me? Yes, great. So I want to say thank you to you, Kasia, for accepting this invitation. It's really nice for us to have you here. Before you start, I just need to give some information for the audience. So first, as I always do, we need to say thank you to these two young scientists here, Ana Carolina and Luis Felipe. They are doing a hell lot of job in organizing this symposium that's held in every week, and it's a lot of effort from them. And thank you again, guys. Also, from today on, we will start to give certificates for people that are attending the talk. And it's really important to say that for you to get the certificate, you need to first watch it live, which means now. If you are watching it at YouTube later, you cannot ask for the certificate. And please, you need also to pay attention in the chat because, let me just do something here. Whatever, yeah. So, because, yes, better. So in the chat, Luis or Ana Carolina, I don't know who is going to do that. They're going to post a link in the chat, and then the people that wants the certificate needs to access it and then fill the form, okay? So, sorry, Kasia, just a moment. For os brasileiros, então, que quiserem o certificado, clique, então, no link que vai ser divulgado no chat e aí preencham o formulário, tá bom? Fiquem atentos ao chat, mas, obviamente, não mantenham, né, a atenção, obviamente, na apresentação. Okay, so the next talk will be from Luciana Pisatti from Universidade Federal do Rio de Janeiro. This talk will be in Portuguese, so sorry, Kasia, I think you're not going to understand anything. But anyway, it's going to be next Wednesday at 4 o'clock. And yeah. And before, so the talk today is from Katarzyna Wojdyla. I hope I pronounce it well. So we met, we got in contact with Kasia when we were in Denmark a few years ago. She was doing her master, then PhD at the University of Southern Denmark. And she did it with Peter Hubstaff. So we all very much know him too. And yeah, it's really nice to have you here. And I know that you learned a lot from him. Today she's enrolled at the Reprogramming Institute, and it's a long story, but her group was from Cambridge and apparently moved to Brabham, if understood correctly. Right, Kasia?
Yes.
So the thing is, I don't know if you forgot the name of this talk, Kasia. It's like my friend, of course, you, and MASSA is the way that we call the mass spectrometry, right? And also it's an adjective when a person, it's like cool. So that's like I don't think this name will work very well for foreigners, but for Brazilians it does. So to show like how cool you are and also like how we are close together. We have some nice moments here. As you can see, this was like a trip that we had together for Rio. So a lot of people from Rio, is attending this seminars. So that's us, of course, doing this picture that everybody go in the Cristo, Jesus the Redeemer. And this is in Fernando de Noronha, cool park, cool place. This is probably the nicest picture that you can have from Willie. And yeah, he's always really very messy. Here you look cute, Willie. Yeah. So without further ado, I want to introduce you give you the talk, Kasia, and please tell us what you've been doing.
Cool. Thanks very much, Tiago, and thank you also to the organizers for giving me this great opportunity to be able to meet up with you guys and to share with you that the work that I've been doing in the last couple of years. So what I'm going to talk about is this work that I've done in collaboration with the lab of Peter Ruggan, and we published it earlier this year. So as you know from Tiago's kind introduction, my background is proteomics, but I really got interested in stem cells. And that is because of the pluripotent stem cells, which I think they are very interesting model to study early human development. And with that comes those really exciting applications and possibilities in both basic and clinical research. So pluripotency is this amazing ability to become any cell of the human body. And it is a transient state that happens very early in the human embryonic development. And as you can see from this slide here, it's also quite heterogeneous because we can have naive cells that come from pre-implantation embryo, and that happens around 6 days post-fertilization. But we also have those primed pluripotent cells that come from post-implantation embryo around E9. And what was always fascinating for me to see is that how very different these two pluripotent states actually are. So we can see that the naive cells grow as this very compact, very tight colonies, and they make this dome-shaped structures. And in contrast to that, primed cells have much larger and much more flat and spread colonies with these very well-defined borders to them. And there is actually more differences. And so in terms of molecular characteristics, we have, for example, in the naive cells, we have global DNA hypomethylation, whereas when they transit to the primed state, the DNA methylation levels increase. Another difference is in terms of the state of the X chromosome. So in female naive cells, X chromosome is active, but then that gets lost when they transition to the primed state. And we also now know a little bit about the surface markers of these cells. So we know from the earlier studies that CD75 and CD100 130 marks the surface of the naive cells, and CD24 and CD90 is specifically present on the surface of the primed cells, and I'm going to come back to that quite often throughout my talk. So the reason why we actually know already quite a lot about these pluripotent cells is because we can now grow them in vitro in the lab using these very well-defined culture conditions. So for example, if we take the naive cells and we grow them in the presence of FGF and serum, they will spontaneously or eventually they will convert to the primed state. And we can also do the opposite. So we can take those primed cells and we can convert them back to the naive state. And this is the process that is called reprogramming. And as you can see from these references here, there has been several different reprogramming protocols already developed, and they basically use different kinds of inhibitors and activators in addition to the basal media that we grow these cells in. So for example, in the case of the 5i based reprogramming, what we do is we use these 5 different inhibitors, and I'm just going to highlight what they do on this cartoon here. So we block Wnt signaling with GSK3 inhibitor. We block MAP kinase signaling with both BRAF and the ERK inhibitors. We also block PI3K signaling by inhibiting SRC, and the integrin signaling by blocking ROCK kinase. So basically, the idea of using all these inhibitors is to kind of block any external signals that come from the outside of the cells and to prevent them from differentiating. But in addition to these inhibitors, we also use the activators. So we use activin A and this cytokine leaf that are there to promote the pluripotent signals. And so what you can see from this is that we have these reprogramming protocols now established, and we use them in the lab, and they work to a certain degree. I mean, maybe the efficiency is something that is still that still needs improvement, but I think the important thing is that we still don't really know what we don't really understand what is happening to all of these different signaling pathways when we transition between these two pluripotent states. And so as you can see, a lot of this, or actually most of this pathway that we act upon during the reprogramming start at the plasma membrane. And so we thought maybe the good starting point for our project would be to use an unbiased technique to analyze cell surface proteome of this naive and primed pluripotent states to kind of try to understand better what is happening in terms of cellular signaling, so the cell-cell and the cell interactions with the environment when these cells are switching between naive and primed state. And so in order to answer this question, we decided to use this method called plasma membrane profiling. And as you can see from this couple of references I've listed here, it has been successfully used in the past to study different biological systems. So the way that this method works is it's based on the labeling of the sialic acid containing glycans in live cells. So basically what we do is that we have our live cells and we treat them with periodate, which would oxidize sialic acids within the glycans. And that makes them accessible for the labeling, which we do using aminoxybiotin. So once our cells are labeled and quenched, we then lyse them, and then we enriched all those labeled proteins using streptavidin beads. And next, we directly digest the proteins that are bound to the beads with trypsin, and that generates the peptides that we use for TMT labeling, and then we can use them in the mass spec analysis, which I'm sure we are all very familiar with in this audience. So I won't spend too much time on explaining how that works. And so basically, this is exactly the workflow that we used in our analysis. So we started with 3 replicates of the naive cells and 4 replicates of the primed. And we did our cell surface labeling, and then we went on with the whole proteomic analysis. And as I mentioned at the start of the talk, we've recently published that earlier this year. And so what we got from this analysis, you can see on this volcano plot here. So this is what is here on the x-axis here is the abundance change between the naive and primed cells, and on the y-axis is significance, and every dot within the graph here represents our cell surface proteins that we identified. So I hope what you can quickly appreciate from this graph is how very different these two pluripotent states are, as almost 2/3 of the cell surface proteins that we identified were either primed specific, and these are marked in red here, or they were naive specific, or naive enriched, I should more say, and they are marked in blue here. And so, because this was such a striking result and so many differences that we saw, so what we wanted to check next was to look for the positive controls. So these naive and primed surface markers that are already known in the literature. And so we were actually quite pleased to see that both CD75 and CD130 we have quantified with much higher abundance in naive cells compared to primed. And sort of along those lines, we also quantified CD90, which is a primed marker with much higher abundance in primed. So that confirmed that that was a kind of a QC of our data set. So next, we really wanted to know what actually are all these hundreds of proteins that we identified in our data set, and to kind of make sense of such a large data set, we decided to take advantage of the Go enrichment analysis. And this is what I'm showing you the summary of on this quite busy graph here. So I'm just going to walk you through it and explain what all of this means. So every circle represents a Go term, and the larger the circle, the more cell surface proteins that they were associated with it. And then the color reflects significance, meaning the darker the circle, the more significantly enriched this Go term was in our data set. And so from this kind of visualization, we can quickly see that, for example, receptor tyrosine kinase signaling pathway was one of the most enriched Go terms in our data set. And indeed, we have identified quite a large proportion of RTK receptors we wanted to have a closer look at this, and this is a example of one of those adhesion-related Go terms that I'm showing you on this volcano plot here. So what I've marked with this purple squares, sorry, is all these cell surface proteins that belong to focal adhesions, and we can see their distribution here. But then what is actually cool about the quantitative proteomic experiments is that we can look a little bit closer and at our data. And this is what I've done here on the example of the integrins. So these are all the integrins that belong to focal adhesions. And this is their quantitation here. So as you can see, the majority of the integrins are naive, are primed enriched, which is what we also saw on the volcano plot here. But there is also this integrin beta 4, which seemed to be more naive specific. And unfortunately, we were not really able to follow this one up because at the time there was not enough there was no good antibodies available that we could use for validations. But we were still quite curious about all these different primed enriched integrins. And so we decided to take a closer look at those. And so the first thing we did is, again, we QC'd our mass spec results with the antibody-based validations using flow cytometry. And this is what I'm showing you on these graphs here. So we can see that integrin beta 1 and alpha 6, they basically stain primed populations much better than the naive ones. And so what we were interested in next was to see if we grow our primed cells on different substrates, whether the expressions of this the expression of these integrins would change. And the reason for that is that, you know, the focal adhesions are these structures that relay the information from the extracellular matrix inside the cell, and so they really kind of responsive to the environment in which the cells are grown in. And so next we set up our substrate assay, and what we did is we grew our primed cells on vitronectin, on matrigel, and the laminin, and then we did the cell surface flow analysis for these two integrins. And as you can see, there wasn't really big changes which we saw with flow, which basically led us to conclude that at least these two integrins robustly stain our primed populations regardless of the substrates that the cells are grown on. The next thing that we wanted to look at was more cell state-specific pathways. And we saw that JAK-STAT signaling was actually significantly enriched in our naive cells. And as you can see from this cartoon here, we identified all these different cytokine receptors that feed into the JAK-STAT signaling, and they were all quantified with much higher abundance in naive cells, which is marked in blue, compared to primed. So this was quite interesting, and the next thing we wanted to check whether high expression of the surface receptors would translate to high activity of this pathway in the naive cells. And to quickly check that, we plotted for phospho-STAT3. And these are the Western blot results of that, which I'm showing you here. So we can see there is much higher phospho-STAT3 levels in naive cells compared to primed, which means that the pathway seems to be more active in the naive state. So next, what we wanted to know what happens to those established naive populations when we perturb JAK-STAT signaling. And for that, we used the pan-JAK inhibitor, so to grow our naive cells in the presence of pan-JAK inhibitor. And as you can see from this Western blot here, using the inhibitor significantly decreased phospho cells, which is something that we would expect. But what we also saw is that naive cells had much lower proliferation rates, when they were grown in the presence of JAK-I. So next we did some RT-qPCR analysis, and we actually saw significant decrease in both STAT3 target genes, which are shown down here. but also quite interestingly, the decrease in the transcript levels of the pluripotency genes. And if we have a look at the KLF4, for example, we can see that the transcript levels for this pluripotency transcription factor dropped significantly as early as 1 hour post addition of JAK-I, and then they were further declined up until 24 hours when we grew the cells in the presence of the inhibitor. And so we concluded that the JAK-STAT is actually needed, the active JAK-STAT is needed for maintenance of human naive pluripotency. And so next thing that we wanted to know is what JAK-STAT inhibition does to the reprogramming. And so to answer that question, we took our primed line and we went and we set up the reprogramming experiment, which I'm showing you on this schematic here, where we did a pulse of JAK-I inhibition between the day 5 and day 10 of the reprogramming. And what we basically saw is illustrated on these images here. And I hope we can all see that by day 12 of the reprogramming, we could see significant decrease in the number of naive colonies in the cells that were reprogrammed in the presence of JAK-I. And we also quantified that with flow. So what I'm showing you on these scatter plots here are the two naive markers that we used in flow. So the double population up here would mark our naive cells, just like we can see them here for the control. And so from that we can see that there was 6-fold decrease in the number of naive cells that we could get from the reprogramming in the presence of the JAK-I inhibitor. And so that led us to conclude that JAK-STAT, that active JAK-STAT signaling is not only essential for the maintenance, but also for establishment of human naive pluripotency. Another aspect to this work was to look for more cell surface specific markers. And this is what I'm going to focus on for the rest of this presentation. So what you can see on this volcano plot here is basically all the surface proteins I've showed you before, but this time I marked all these proteins that we thought are potential interesting new candidates for new markers, and they are marked in black. And I've also marked our positive controls. So the reason why we selected those proteins is because they had large fold changes from our PMP analysis, and they also had corresponding data from transcriptomics in both cells and the human embryos. And also more practically, we, at least in the first instance, we went for all those candidates that we could get good quality flow antibodies in order to do our validations. So as you might guess now, what we did next is the flow cytometry based validations, and these are the results of them here. So on the left-hand side are our positive controls, so the naive markers up here and the pride markers down here. And this is just a selection of a couple of our candidate new naive markers. And so we can see that that most of them can nicely separate our naive populations, which are marked in blue, from primed populations, which are marked in red. And I hope we can all also appreciate that we get a similar kind of shift between the two populations from these new markers like we do for our established markers. We also checked for their distribution using immunofluorescence, and this is what I'm showing you on this picture here. So in the red is the self-staining of our candidate new markers, and as we can see, they are nicely staining the periphery of the naive cells, but there is no or very little signal that comes from the plasma membrane of the primed cells. And so the next thing that we wanted to do, well, we did actually for all these promising new candidates like SASD2, we really wanted to be sure that their surface expression is not just specific to one naive line. And so we did flow analysis where we checked for SASD2 surface expression in both H9 cells, and these are the cells that we used for our original screen, but also in WIBI3 line, and also in and we also used cells that we derived from several different reprogramming methods, which I'm showing you all here. But basically, the take-home message from this analysis here is that SASD2 could nicely separate our naive population, regardless of the naive cell type we used or the culture conditions that the naive cells were grown in. What was also nice to see is that when we checked published transcriptomics data sets from the human embryos, we could actually see high expression levels of SASD2 at this early day of the human embryonic development, which I showed you before corresponds to the time frame where this pluripotent state actually happened. And so we were actually quite excited to think that we maybe found a new naive markers that will be a robust protein that we could use to mark our naive cells. And actually, we were not the only one who came to that realizations, and as our colleagues from the Stem Cell Institute also independently showed similar data and reported SASD2 being a robust naive markers a little bit earlier than us. But still, nowadays we actually use SASD2 routinely in our lab, and we use it for the quick flow experiments where we check the purity of our naive cells. So what I'm showing you here is the original panel that we used previously, which contains all these 4 different naive markers. But what we can actually do now is we can swap these 3 for SASD2, so have basically double staining for SASD2 and CD75 positive populations, and still be able to confidently isolate those naive cells that come from the reprogramming experiments. And the reason why this is useful is basically a practical one, that having just 2 markers removes the need for the fluorescence compensation, and so it's much easier to do this kind of panel, especially for the new people that join the lab and maybe are not so familiar with the multicolor flow analysis. Next, I'm going to talk a little bit about FOLR1, which as you remember from the previous slides, was also one of the potential new naive markers. And this is actually the one that we were able to show some signal in the human blastocyst. So this is what I'm showing you on this picture here. We can see some FOLR1 positivity that comes from the inner cell mass of the human blastocyst. So this was nice to see, and we really were excited to have additional naive marker, but we also wanted to learn a little bit more what FOLR1 does in naive pluripotency and what its role is. And for that, we took advantage of the CRISPR-Cas9 system, and we removed this exon 5 of the FOLR1 gene to generate FOLR1 deficient prime cells. And the idea behind that was that once we have our FOLR1 deficient prime cells, we can then use them in reprogramming experiments to generate naive lines, or to see if we would actually be able to generate naive lines. And this is actually the work of the former PhD student in the lab, Mandy Collier. And so what Mandy did is she took those FOLR1 deficient prime cells and she took them through the course of the 5i based reprogramming, and you can see the different stages of that on these photos here. And so what we can see that we actually were able to see naive colonies as early as day 10 of reprogramming, and they were then further purified up until the point where at day 34 we had basically very pure naive line that was FOLR1 deficient. And so from this experiment, we concluded that FOLR1 is not really essential for naive cells to be established in the course of reprogramming. But having this new line in the lab, we wanted to characterize it a little bit more. So the first thing that we did is that we, of course, checked whether we actually removed FOLR1 from the surface, and this is what I'm showing you on this histogram here. So we can see there is no FOLR1 positive signal coming from the knockout cells compared to the controls. And we also confirmed with the Western blot that the protein is actually gone. So the next thing that we look at was whether FOLR1 deficiency affects the expression of the pluripotency genes. And these are the results of that here. So basically we can see that the majority of the key pluripotency genes were not really affected by loss of FOLR1. However, we could see small but significant increases in both OCT4 and KLF4 expression. And so because we didn't really see a clear phenotype coming from the reprogramming experiment, we turned into FOLR1 function to try to understand its role in the pluripotency. And what FOLR1 does is part of the family of these folate receptors that they basically take up folic acids and other folate derivatives, and then they take them inside the cell where they are used in many metabolic processes. But what I'd like to highlight here is that one of the outputs of these processes is that it generates methyl groups for methylation of both DNA and proteins. And so the next question that we asked was, does FOLR1 deletion affect global histone methylation levels in the context of its function? And these are the results of the analysis that I'm showing you here. So basically what we did was we blotted for several different histone marks in our FOLR1 deficient cells in the parental controls. but we were not really able to see any significant differences in either active or the repressive marks. So because there was no differences in protein methylation levels, we then went on to check what happens to the global DNA methylation levels. And this is what I'm showing you on these graphs here. So we can see that also there was no significant change in methyl C and hydroxymethyl C between our knockout cells and the controls. So this was a little bit disappointing, but perhaps not all that surprising when we then went back to our data and we could actually see from our PMP screen that in addition to folate receptor, there are also several different folate transporters which are expressed on the surface of pluripotent cells, and actually some of the transporters are quite abundant on the surface of naive cells. And then we also checked for the expression of other genes from the folate receptor family using RT-qPCR. And as you can see from these graphs here, there was a significant increase in the FOLR3 expression levels in our knockout cells compared to the controls. And so all that might provide some form of evidence for the compensation that is happening in these cells in the absence of the FOLR1 receptor. And so just to sum up, I've just presented you the data from our plasma membrane profiling experiment, where we quantified changes in cell surface proteomes of naive and primed human pluripotent states. We identified new naive cell surface markers such as FOLR1 and SASD2. We also showed that the active JAK-STAT signaling is essential for both establishment and maintenance of naive cells, and also we've generated this large resource that we hope can now be used in the community to and be interrogated to extract new biological hypotheses for the future projects. And especially with that in mind, we also generated this web resource, which I think I should have a print screen of. So all our data from the PMP analysis is available via this link here. And this is just a print screen from the website. So basically, the data is fully searchable using this search bar function here, where one can search for their favorite genes or proteins, or even for a specific Go term or other keyword. And then the results of the search are populated in this list here. And then when you click on the individual proteins, that populates the graphs that with the quantitative information, which can be directly extracted into PDF or JPEGs and used in presentations or for grants, for example. And so with that, I would like to thank everyone who was involved in this project. So first of all, people at the Abraham Mass Spec facility where this project originated, and especially David for giving me the freedom to pursue my research interest. But really big thank you goes to Peter and the whole RAPGAN lab for basically adopting me in their lab and showing me all these cool techniques and the human ears culture that I got to learn during my stay there. And also many thanks to the Abraham Institute facilities, especially to Laura, who was the person behind our web resource. I would also very much like to thank the Lehner Lab for sharing their expertise on the plasma membrane profiling, and Frederick and John for their help with the human embryo stains. And this project was supported by the funding from BBSRC. And so I'll stop here, and I would, of course, very much like to thank you for your attention, and I'm happy to take some questions.
It's better to unmute the microphone, right? So thank you very much, Kasia. Really interesting this research and what you've been doing. Before, we have like a kind of 10 seconds delay between what we are talking here and the people in the YouTube, so probably they're seeing the end of the talk now. But anyway, I will start making questions. As here, it's a source for information for a lot of students, and I know that a lot of people are interested in membrane proteome. I would like that you comment a little bit more about this approach that you use, right? This, if I understood correctly, you target the sialic acid to modify it, and then you use biotin you biotin it, and then avidin to fish them out, right? So what it's like advantage and disadvantage of this approach, for example, compared to a simple shedding, which would be pretty much like just like chop up all the proteins that are facing extracellularly by trypsin, for instance. You know what I mean? Like a shed. Yeah, I think it's shedding. That's the name that you call it. And yeah, if you can comment on that, and also what kind of softwares you use it for your data analysis. For example, I really enjoyed. I know that Willie the Grumpy, he doesn't like very much gene ontology. I don't know if he changed his mind.
Uses it
but I
to annotate his cell surface proteins.
Yeah, he likes to complain, but he uses. I know how he does. Yeah, it's grumpy old man, and he's getting bold, by the way. And but what about just here? Yeah, but what about the software that you use it to create this very cool plot that you use it to show your gene ontology data?
So let me start with the graph, because I'll start from the back side. Go is a waste of time. No, he should not be allowed to comment. So yeah, so what I use these days for Go enrichment is the software called Enricher. I don't know. It's called Enrich R. So it has a web-based interface, just kind of like the normal Go terms, but basically it contains Panther and KEGG and all the other geo search tools in one place, and it's also that the most up to date is quite frequently curated and managed in a way that actually things are updated, unlike some other tools. So just to get the Go terms, I used Enricher, and then I used this software called ReviGo. And that basically what it does, you put all your Go terms into ReviGo, and then it kind of groups similar Go terms to the same categories, so kind of filters out the data set, and it makes like a summary that you can then input to R, and you can generate this plot that I showed you. So that's basically how it's done. And regarding the method, I'm not really an expert on that, so I'm just gonna say what I know. Basically, I guess the advantage of this method is that it um, it's it's specific in a way that it will only enrich, the proteins that have glycans and that have sialic acids within the glycans. So that the advantage of that is that there is less background in your data set coming from non-specific stuff because you specifically enriched for the glycosylated proteins. The limitation.
And do you deglycosylate? Sorry, do you deglycosylate before digestion, or you later you analyze everything?
No, you no, I don't deglycosylate.
Okay.
Because, well, I suppose you could do that later, but at the enrichment step, you really need that glycan parts in order to pull out the labeled proteins and to enrich them. Yeah, about the shedding method. Well,
They were actually, they were correcting me. It's shaving. Yeah, it's like I need to do here, by the way. It's shaving. Yeah.
Yeah, I'm not. So you mean just to chop it off at the surface with glycans and at the surface with trypsin and then just analyze that?
Yes.
Yeah, I don't.
Marcela has a question for you, I think. Can you read it?
Right. Seems that the naive have more sialic acid than the primed ones. Do you think that sialic acid would improve the implantation? Huh. I don't know. I didn't measure that. So basically, yeah, I think that's the fair thing that I can say at the minute, that we didn't specifically profile the glycans. We just kind of relied our well, we rely our analysis on the fact that mammalian cells are glycosylated, and we don't expect to see differences in glycosylation levels between naive and prime, but that might be an oversimplification. It might actually be that there is a difference between the sialic acids in these two states. I don't know.
Great. Willie is also, he wrote something here, don't know. Okay. Thank you. Great talk. Yeah, Gabrielle has a question for you.
Okay, that one I think I will be able to answer. So the question is, what is the advantage of labeling sialic acids instead of primary amines, such as the side chains of lysins, as the classical biotinylation protocols for surface-some proteomics? So I think basically it's what I've mentioned before, when that there is lower background, there is lower contamination from intracellular proteins when you label them via glycans, because the NHS ester can basically enter the cells and then label intracellular stuff, and that would increase your background of the analysis.
Simone as well.
Can you do PMP after you sort cells by FACS? You could use TMT to boost the sensitivity. Maybe you do that already. Sorry, I did not get it. So yes, you can do PMP after sorting, as long as you sort, yeah, as long as you don't fix your cells. because once they fix, once they fixed, you risk basically getting labeled stuff, intracellular stuff. So if they're not fixed, it should work. And you could use TMT.
What's the number of cells that you use to label?
So it was 10 million per condition. You can go lower. You can use 5 million. You can go up to 1 million per condition. But anything lower than 1 million, you sort of starting to having issues with the recovery of your proteins. So I think 10 million is a good starting point.
And she is also asking one more.
Pluripotent cells can be very confluence sensitive, self-differentiating in high confluence. Did you see differences in your response to JAK-STAT related proteins at different well confluence? We did not check for that. I mean, as you pointed out, these cells really don't like to be either too sparse or too dense. So we just try to keep them at the happy density, and then we did our proliferation curves at the densities that were optimal. So we didn't really look, what's the response of it when they are over confluent.
I think Simone wrote something else here. Right.
Right. That's why I was wondering. That's why I was wondering if you can use a TMT channel as carrier, like people do for single cell proteomics. I suppose. I mean, I think if you can afford to have, I mean, if you can afford to get 10 million cells per condition, I think you have enough to not have to need to do that. Oh wait, but then they're talking more. The fixing can be an issue.
Are you reading another one?
It was a reply. I think they're just having a chat with James.
Yeah, both were complaining about the gene ontology. Whatever.
Yeah, I mean,
It's really a pain. Yes, I agree with them. That's like most of the time useless. But sometimes you send a paper, and then the reviewers will ask you, "Where is the gene ontology?" So it's really. I think it's.
I think maybe it just depends how you use it. Like,
Yeah.
if you just use it to kind of visualize your data and to give you directions for more like functional work, I think that can be informative. Maybe the problem comes when you just publish the list and you put the go terms, and then you don't really. I mean, that there is no follow up to validate what gene ontology is saying in a way.
Yeah, Marcel is saying here that she likes in the start. I agree. It's like maybe it's a way just to start surfing on your data sets, and yeah. So yeah. Simone has another one here. Normally, what?
Yeah, I think yeah. So basically, what Simone, I don't know. Should I read the question or should I just answer? I they haven't been the same starting batches. I think it's just kind of takes the practice to be able to handle them and to keep them happy, as we call it. And once you kind of get a hang of that, you can grow them in a reproducible way. But yeah, it takes quite some practice.
Yeah. Let me just make an announcement in Portuguese. Sorry. Pessoal, vamos colocar o link aqui, tá? Para vocês assinarem. Deixa eu ver se eu vou conseguir aqui, ó. Eu acho que foi. Yes. Okay. Sorry. Yeah. It's for the certificate. What else? I don't know if I jump at some question. So in your lab, do you do a lot of stem cell? People there do a lot of stem cell work, or it's more like a method development, or what's like the main interest of the group? Like the main question?
I think that the main question, well, it's basically the epigenetics lab where I used to be. So the main questions are related to how the whole genome and epigenome is regulated in these early stages of development. So it's a lot to do with, like activation and silencing of genes from the X chromosome, and also looking at the enhancers and promoters and all the whole like gene regulatory network, how it is all rewired in these early stages of development. And yeah.
Okay. I think that all the questions were answered. So I want to thank you again, Kasia. It was really nice to see you. I think that almost 2 years that we didn't see each other, like at least over a screen. And yeah, thank you very much. It was really, really cool.
Thanks.
And you take care. You and Willie.
You too. You too.
So,
bye-bye.
Yeah, Simone is saying here, epigenetics rules all. Oh my God, okay. Marcel is asking something else, is it? So Marcel is late. Marcela, the event is nearly ended.
It's Chris, we can take it up. We can take it up in a different channel.
Yeah, you can answer. It's yours.
Well, I think she said it herself. I mean,
the Christmas one surely for this other set. Yeah, that's true. It's not a, sorry, my fault. It's not a question. So you both take care.
You too.
Bye bye, guys. Bye bye.